Structure of chromosomes. Each chromosome in its condensed form, as visible during the start of the cell division, is made up of two chromatids. As they are similar to each other, we call them as sister chromatids, which are joined at one point, and that point of attachment of these two chromatids is called as centromere. So this is the structure of chromosome. These are the chromatids. So two chromatids are present in each chromosome. As they are similar to each other, they are the part of the same chromosome, we call them as sister chromatids. And these sister chromatids, they are present, they are joined at one place. The point of attachment of these chromatids is called centromere. Now location of centromere in each chromosome is of particular type. According to the position of this centromere in a chromosome, chromosomes are of 4 types. Now the 4 types of chromosomes according to the position of centromere are, the first one are metacentric. Meta means at the center. So if the centromere is present just at the center of two sister chromatids, like in this diagram, that type of chromosomes are called as metacentric chromosomes. And if this centromere is present slightly away from the center, not exactly at the center, little bit away from the center, then the chromosomes are called as submetacentric. Now the next are acrocentric. Means if the centromere is present towards one end of the chromosome, or one that is not exactly at the end but nearer to one end of the chromosome, then the chromosomes are called as acrocentric. So in acrocentric chromosome, one arm of the chromatid is long, very long, and another is very short. And last one is telocentric. Here the centromere is present at one end. At one end the centromere is present, and that is called as telocentric. Now this centromere is also very important during cell division. During cell division, spindle fiber hold the chromosome, and then with the contraction of the spindle fiber, these chromatids of the chromosome, they separate out and equally distributed in resulting daughter cells. So centromere is not only the point of attachment of these two chromatids, this is the point where spindle fiber get attached to the chromosomes during cell division, and when those spindle fiber get contracted, they results in the division of these two chromatids. They results in the separation of these two chromatids during cell division. Once the chromatids are separated, they are equally distributed in two daughter cells which are formed after the divisions. On getting inside the daughter cells, these chromatids, they decondense themselves, and when they decondense, again they form very thin thread-like structures known as chromatin fibers. And again those chromatin fibers which are present in daughter cells, when those daughter cells prepare themselves for the cell division, again the chromatin fibers, they coil, they get condensed to form chromosomes for the next cell division. So this is all about the structure of chromosomes. Now next we will start with the progress check, which is given on page number 11. Now progress check given on page number 11. Fill in the blanks. Chromatin fiber is made up of DNA and histones. Second, the two sister chromatids of a chromosome are attached to each other at centromere. Third, the structure of DNA was first discovered by Watson and Crick. Next, The three components of a nucleotide are, first is phosphate, second is sugar, and third nitrogenous bases. Fifth, DNA strand wound around a histone octamer, 8 histone molecules, form a complex called a nucleosome. After this, a specific sequence of nucleotides on a chromosome constitute a gene. One topic I forgot, that is what are genes. So let's complete it also. What are genes? Genes are specific sequences of nucleotides on a chromosome. Or in a simple words, genes are the segments of DNA because nucleotides are present on DNA. So genes are the segments of DNA present on the surface of chromosomes that encode particular proteins which express in the form of particular feature of the body. They help in expressing particular characteristics present in our body. They are the unit of heredity. They are the unit of heredity which transfer from parents to offspring. They help in transferring hereditary characters. The characters which are present in parents, they are transferred to the next generation through these genes only. And these genes are present over the surface of chromosomes. Now, one extra information not in syllabus, that is DNA fingerprinting. Lot of reason in between genes are just non-functional. Now, genes are present over the surface of chromosomes. So, in between the genes, there are regions which are non-functional. Such areas, about 99% of the total DNA, shows tremendous variation from person to person. These areas, they are unique for each one of us, and they help in identification of the individual by what is popularly known as DNA fingerprinting. DNA fingerprinting is useful in establishing paternity and maternity in criminal cases. They help to identify the criminals at the crime scenes. Blood or semen stains, hairs or items of the clothing found at the site of crime provide biological evidence through DNA fingerprinting. So it is very important in investigating the crimes or at crime scene. Next, we will start with need for new cells.